thank you everyone. Again, let me start again what we need to know for reduction division. So we have these three learning outcomes to be achieved for our class now. So first thing that's you need to explain why we need to have the reduction divisions, reduction division, or commonly known as meiosis, before the fertilization in sexual reproduction. Only for sexual reproduction. Asexual, no need. Asexual is nothing to do with meiosis, but more to the mitosis. Okay, and then we need to describe with the aid of photomicrograph. Question always asks, highlight this, behavior of the chromosome. Question always asks about this rather than, okay, even though we need to know, but quite less chance to come out. For example, the behavior of the nuclear envelope, cell surface membrane, spindle. Okay, and in my notes, I will add in few more things. Okay, for example, the behavior of the centrosome in the centrosome. Okay, in animal cells, centriole, for example. Okay, we're going to talk about the behavior of the nucleolus also. Okay, as the extra things that we're going to learn. Okay, then during the meiosis, very, very important, because meiosis actually do lead to this genetic variation, do give us genetic variation. How? Okay, the mechanism is by crossing over that take place during prophase 1. And also the random assortments, or we call independent assortments of the homologous chromosome that take place during metaphase 1. Okay, so these two events actually can give us the genetic variation. But this is due to the meiosis, random fusion of the gametes. Now, what means random fusion? Because when the fertilization takes place, for example, in humans, okay, so when male ejaculates the sperm cells, maybe about 2 million to 3 million of the sperm cells, we have no way to choose which sperm cells, and each sperm cells represents a combination of the allele. Okay, so same goes to the female. Each month when you release the secondary oocyte, or we call as the ovum, also the same thing, because you can't choose which ovum to be released. So this random fusion, because only one sperm cell can fertilize one female gamete, therefore it will allow more or higher genetic variation to take place. And we haven't counted the probability your father met with your mother. So it makes us, each single of us, very, very unique. Okay, so one of these for random assortments or independent assortments, I will do a simple calculations and you will know how unique or how special each of us is. Okay, okay, so why we need to have a reduction divisions? So two things we always talk about this. Okay, summarize here. Okay, so sometimes question will be 3 marks, 2 marks, or we ask about the need for the reduction divisions. First thing, we want to reduce. Reduce what? Reduce the number of chromosomes. Okay, reduce the number of chromosomes by half, or half the number of chromosomes. Okay, so that we will be able to make the diploid cell 2N become haploid N. This important point, first point, okay, first part. So we want to reduce the number of chromosomes by half and make the diploid cells form haploid cells through this meiosis process. Okay, so why we want to do this? So that when during fertilization, we will be able to restore back the original diploid number. Okay? Restore back diploid number, the original diploid number. If we don't do this, what will happen? If we don't do this, a bad thing here is we want to, because we restore back already, then we can prevent. Okay? So if we don't do this, what will happen? What's the consequence here? We will have a doubling up of chromosome number for each round of the fertilization. So these 4 key points that you have to remember. Okay, why we have or why we need to have reduction division? first we want to reduce the number of chromosomes by half and make or to form the haploid gametes from the diploid cells. Okay, and for fertilization take place, fertilization of two haploid gametes will be able to restore back the original diploid number and prevent the doubling out of the chromosome number in each fertilizations. Okay, so highlights the first part here, my notes. This is very important for you guys to answer the questions when the question asks the needs for the these reduction divisions. Okay, so the type of the nuclear divisions that halve the number of chromosome is termed as the meiosis, and meiosis also introduced genetic variation. Okay, meiosis also. Okay, so we're going to learn it when we come to the third part. So this diagram summarizes the process of the life cycle involving the chromosome number, whether at a double or half the number during the meiosis. So what are the stages of meiosis? Okay, so it's the same it's almost the same as the mitosis. So we're going to start it with the interphase, followed by the meiosis 1. So in this case, you have the PMET. Okay, prophase 1, metaphase 1, anaphase 1, telophase 1, and then continue with the meiosis 2. Again, we're going to have the P1, P2, metaphase 2, anaphase 2, and also telophase 2. And then in between meiosis 1 meiosis 2 depend on the kind of the organism. Some may or may not have the cytokinesis, but after meiosis 2, then cytokinesis take place. Okay, so interphase basically where the DNA replication will take place, organelle replications. However, the single DNA replication is followed by two consecutive nuclear divisions, okay, which term as meiosis 1 and meiosis 2. So after each these two time of divisions will result with 4 daughter cells, okay, each with only half as many chromosomes as the parental cells, or we call it formations of the haploid cells. Okay, so let us look at the process, okay, in more detail now. Okay, now very simple quick ones, very simple quick one. If I have a 2n equal to 4, this is the diploid cell. 2n equal to 4. So at the end of the meiosis, I should get, or I must get the haploid cells, which is n equal to 2. Okay, n equal to 2. So if I start it first, let's say in G1 phase, interphase G1, this is my cell. Okay, 2n equal to 4. So I have the blue one, I have the red, they are a pair. Blue and red, long chromosome, so they are a pair. Green, short one, and orange. So can you see that? One long, one short, one set. Red and orange, one set. Blue, green, one set. So in this case, I will write here number of chromatids equal to 4. Each chromatin in this case, because haven't replicated yet, right? So number of DNA here also equal to 4. Each chromatin consists of 1 DNA. So are you clear at this moment? Number of chromatins equal to 4. Number of DNA also equal to 4. Okay, yeah. So after S phase, so what actually happened? What's the events that take place during S phase? DNA replication take place. Okay, DNA replication. So means that what you're going to see here is from one blue color chromatin, you're going to get the second blue color chromatin because DNA replicate semi-conservatively. Okay, this is very important because of semi-conservatively, so we get the identical DNA. So after the S phase, what you expect to see? So you expect to see the cells here So blue color will form like this. Okay, red color, and then green color, and orange color. So if I ask you now, what is the number of chromatins? Okay, the number of chromatins here were equal because they are still together. Chromatins, can I say they join at the centromere there, right? So number of chromatins still equal to 4, but number of DNA, because we replicate the DNA now, we replicate the DNA, so number of DNA equal to 8. Can you see that? Blue got 2 long one, red you have 2 long, okay, green you have 2, and also orange you have 2. So number of DNA equal to 8. So this basically something happens during the interphase. So once everything ready, so then the cells are going to proceed into the meiosis, which is the prophase 1. Okay, so now let us look at the prophase 1. This meiosis 1, prophase 1. Prophase 1 actually quite busy. A lot of things actually happen during prophase 1. Okay, so first thing we're going to look at here. I'm talking about behavior of the chromosome first. Behavior of the chromosome. So first thing, what actually happened here is condensations of the chromatin. Okay, first event. Condensations of chromatin. Now it takes time, so it's not like magic. Okay, after 1 second they become thicker already. So during the condensation of chromatins, then you can see that homologous chromosome actually move toward each other, and we call it as a homologous chromosome pair. Are you clear? Okay, homologous chromosome pair, because protein-protein interaction allowed they recognize each other, then they start to move closer to each other, pair up. So once they pair up, they can carry out what we call the crossing over. So crossing over is the one that actually gives us the genetic variation later. I will explain in detail later. Okay, so when crossing over already take place, they join together. The homologous chromosome join together. So they join at where? They join at chiasmata. Okay, they join at chiasmata, forming what? Forming what we call the bivalence or tetrads. Let us say bivalence or tetrads. I will show you guys later. Okay, what does this mean? Okay, so basically what will happen is the blue one and the red one, they are homologous. So when they are homologous, so they will do what we call the crossing over. Can you see that? So this is what I mean by the chiasmata. So they join as the chiasma singular, chiasmata plural, right? Plural. So the entire thing here now is termed as the tetrad. Okay, named as tetrads or bivalence. Okay, either tetrads or bivalence. Okay, so if you zoom it in, you will know that they do have the centromere. This centromere, and centromere will be attached to the spindle microtubule. Okay, but you can see that compared to the mitosis, mitosis is from 2 sides, but this one only from 1 side. Each of them only from get 1. So this is a spindle microtubule or spindle fiber. Okay, spindle microtubules. Okay, so push each other until later stage in metaphase 1, they will align at the metaphase plate. Okay, so I'll go through again with you guys. So condensation of chromatin. So during the condensation of chromatins, this homologous chromosome now pair up. Okay, homologous chromosome pair up, and then they start to carry out crossing over. So this will give us genetic variation. So what actually happened during the crossing over, I will tell you guys later. Basically, they exchange the DNA material. Okay, or more precisely, they exchange the alleles. Are you clear? They exchange the allele. Okay, so then when they exchange, but they haven't break up yet. In this case, they join together at the chiasmata, forming the entire structure. We call them as a tetrads or bivalents. So you're going to draw it here. You will see something like this. Okay, the blue color. And the red color. They're homologous, guys. They're homologous. Then green color. And also the orange color. Okay. So in this case, guys, if I want to ask you, what is the number of chromosome? Okay, because now we can call it a chromosome already. Number of chromosome. So one red, one blue, one green, one orange. So equal to 4. Okay, but now each of them have 2 sister chromatids. So number of DNA molecule now, or sister chromatid here, will equal to 8. Okay, so the rest of the events that remain the same, guys. For example, centriole or centrosome, centrosome start to move to the opposite pole. Nucleus, okay, disintegrate. Nucleus disappear. Nuclear envelope disintegrated. Still remain the same. So I only pinpoint out the behavior of the chromosome. Okay. So from here, now next stage. So due to, okay, this is centromere, 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 centromere. So formations of the spindle microtubules. Oh, sorry. Okay, formation of spindle microtubule. Formation of spindle microtubule will push them. Can you see that? Try to imagine that someone push them. So push them to where? Push them to the metaphase plate. So we'll come into the metaphase 1. When during metaphase 1, what you expect to see? A signature event. So in metaphase 1, you're going to see that these tetrads or bivalents align at the metaphase plate. So you can see that the blue one and the red. Can I see that? Okay, the blue and the red, then greens with the orange. Can I see that? Okay, so what they align? Where they align? They align at this something like an imaginary plate here. Can I see that at the center here, right? So we say that first, the tetrad or bivalents aligns at the metaphase plates or equator of the spindle. Okay, it's the same thing. Okay, tetrad bivalents align at the metaphase plate. Or sometimes you can use the words equator of the cells or equator of the spindle. Okay, and if you zoom it in, then you realize that now all of them will have the spindle microtubule attached to the centromere. Okay, so we have the spindle microtubule attached to the centromere. Can you see that? So because of this, okay, then they will be able, okay, to align at the metaphase plate. And if in the animal cells, only in animal cells, this one, then you can see that the centrosome or centriole or centrosome, centrosome, okay, reach at the opposite pole of the cell. Can you see that? So these are the 3 important events that take place. Okay, now here we have a little bit of a special case take place here. Now, what means special case? We do have this what we call the random assortments or independent assortment take place, which create the genetic variation. Now, what's been independent assortments? If you understand the concept of the meiosis, at the later stage, you will see what? Okay, during the anaphase. Okay, during anaphase, okay, look at my cursor here. We are going to break anaphase 1, we're going to break the chiasmata, break the chiasmata, break the chiasmata, break the chiasmata, and then the blue color with a little bit of red will move this side. Green color with a little bit of orange, move to the left. Red color with a little bit of blue, move to the right side. Orange color with a little bit of green will move to the right side. But this, you can see that if you divide them into two cells, when you divide them into two cells, if the blue with the green will be together, red and also the orange will be together. But this, we assume that they arrange this way. If, let's say, the arrangements of the chromosome is like this. Because they are random. In this case, the arrangement are random. So it means that you're going to get a different combinations. Can you see that? This one will give us a different combination. And this will give us what we call the genetic variations later. Okay, so I will tell you guys later, in more detail about the independent assortments together with the crossing over. But at this moment, you can see that how the cell actually, how these are tetrads or the bivalents position themselves will result in the formations of the genetic variations. Okay, so after metaphase, now look at metaphase. So again, number of chromosomes still equal to 4. Number of DNA per cells still equal to 8. Nothing changed. Nothing happened to the number of chromosome. Nothing happened to the number of DNA. They just align at the metaphase plate. Okay, so after metaphase 1, the cell will progress to the anaphase 1. So in anaphase 1, what you expect to see, because of the shortenings of the spindle microtubules, okay, shortening of the spindle microtubules. Now you can see that I use the first options. So blue color chromosome, okay, with a little bit of red color, okay, and then green color chromosome, with a little bit of the orange. Okay, here we have the red color chromosome, with a little bit of blue, and then we have the orange color chromosome with a little bit of green because of crossing over. Okay, so what they do, they're going to move towards the centrosome, away from the metaphase plate. Okay, so what is the event take place? Now be careful. First, we have the shortening of spindle microtubule. So when the shortening of spindle microtubules take place, you're going to break. Now, be careful, guys. The correct term we use here is to break the chiasmata. Sorry, I need to highlight. Break the chiasmata. And then when they break the chiasmata, it's not a sister chromatids. The entire unit, can you see that this one, they are still chromosome. So chromosome move? Move to where? If they say animal cells, okay, animal cell, we say move to the centrosome or centriole. Away from who? Away from the metaphase plate. Okay, so question always like to ask you guys to draw particular anaphase. Particular anaphase, why? Because anaphase 1 and anaphase 2 totally different. Okay, anaphase 1 and anaphase 2 totally different. You can see that now. Okay, you can see that now. That is the break of the chiasmata. And then who move to the opposite pole? Okay, is the chromosome, not sister chromatids. Chromosome is the one. Okay, move to the centriole, or move towards the centriole and away from the metaphase plate. So if I ask you here, number of chromosome still remain 4. Number of DNA per cells, because how many sister chromatids? Total 8 sister chromatids. So therefore, we have 8. And number of DNA will be 8 or sister chromatids. Okay, so if we proceed to telophase 1 and cytokinesis, then you can see that the cells form the cleavage furrow. Nuclear envelopes start to reform. Correct or not? Okay, so we can see that now here you have the blue chromosome with a little bit of red. And then you have the green chromosome with a little bit of orange. This side, you're going to have the red, blue, and then you have this orange color chromosome with a little bit of green. Okay, so this is telophase 1. So during telophase 1, what will happen? So we say that chromosome reach the opposite pole. Opposite side of the cells, okay, opposite end or end of the cells. Okay, and then when they reach, then it will uncoil to form chromatin. Here, behavior of the cells. So what you expect to see later? Cytokinesis take place. Okay, cytokinesis take place, then you'll see that now I form 2 cells. After cytokinesis, 2 cells. So because they are uncoiled already, right? They are uncoiled already, so you can see that the blue color with a little bit of red. Green color. Oh, too big. A small chromosome, sorry. With a little bit of orange. Then we have the orange chromosome. With a bit of green. And you have the red chromosome. Oh, my bad. With blue. Okay. Now, in this case, guys, be careful. If I ask you number of chromatin per cells, can you see that number of chromatin per cells? 1, 2. Can you see that per cells? So means that after meiosis 1, it's very important, meiosis 1. The function of meiosis 1 actually is to separate or segregate homologous chromosome. So means that at the end of meiosis 1, you already get haploid cells already. Correct or not? Number of chromatins per cell is equal to 2. But if I ask you number of DNA per cell, is still 4. Correct or not? Because you have 2 sister chromatids per chromosome. So therefore, we still need to continue the meiosis 2. So what's the functions of meiosis 2? If I ask, you put it here, the functions of meiosis 2, now why we need to continue. Meiosis 2 almost equal to mitosis. All the events almost equal to mitosis. The idea here is to segregate, means to segregate sister chromatin. Are you clear? And the cell is still be haploid after meiosis 2. So the idea, the role of the meiosis 2, basically we want to segregate the sister chromatids. Okay, so means that at the end of the process, I don't want to equal to 4 anymore. I want it to become number of DNA per cells equal to 2, because number of chromosome equal to 2, number of number of chromatin per cells equal to 2, then number of DNA per cells must equal to 2. So that's why we have no choice but to continue, okay, to continue to carry out the meiosis 2. But slightly special about meiosis 2 because before meiosis 2, we don't have interphase anymore. Are you clear? After cytokinesis, they continue with the prophase 2. Okay, so now we go into prophase 2. So event also the same thing. So we have 2 cells now. So I need to take care of these 2 cells. So here we have a blue. Okay, can you see that? They already coiled in or condensed into the chromosome. And then we have the green with a little bit of the orange. Then I have the orange, a bit of green, and I have a red one with a bit of blue. Okay, so can I see that? So again, if you look at this number of chromosomes per cell, equal to 2, but number of DNA per cells still equal to 4. Okay, so what is the signature event? Similar to mitosis. So we say that first, okay, chromatin condense. Okay, chromatins condense into chromosome. Okay, let's see that. This is the signature events of the prophase 2. And then continue to the metaphase 2. So during metaphase 2, then you can see that again, the chromosome now aligned at the metaphase place, similar to the mitosis. So you can see that the blue color, a little bit of red, and then green color with a little bit of orange. Okay, and don't need to worry about how to copy or not, because it's in video form, so you can actually always check it back. Great. Okay, and if you check, you know that at this moment, centromere attached to the spindle microtubule. Can you see that? Okay, this metaphase 2. So what are the signature events? So we say that this is chromosome. So chromosome, okay, chromosome align at the metaphase plate or equators of the cells. Okay. Now, next, number 2, what actually happened here is centromere are attached to spindle microtubule. Yeah, it's attached to spindle microtubules. So what is the next thing happen? Next thing happens for metaphase 2, then you're going to have the anaphase 2. Now, anaphase 2, you guys need to remember. Anaphase 2, you guys need to remember. Okay, so in this case, you can see that the cells so you can see that this, because of the shortening of the spindle microtubule. Okay, shortening of spindle microtubule. Now, this is very important. We use the word divides the centromere. Highlights the words divides centromere. Don't use the word split or break. This under CIE, you cannot use the word split or break. It must only one word to be used, divides the centromere. So when I divide the centromere, because of the shortened spindle microtubule, then you can see that now sister chromatid is the one that moves away. Right? Sister chromatid move towards opposite the end. Okay, move towards opposite end, away. Okay, away from the metaphase plate. So how it look like? How it look like, huh? So you can see that blue color, green color. Okay. So blue, a little bit of red. And then green with a little bit of orange. Can you see that? Here, so move. And then here you will see a red color. And then red, with a little bit of blue. You got the crossing over, right? Then here you have the orange. And you have the orange with a little bit of green. Okay. So the correct words to be used here is the sister chromatid. So this is the sister chromatid. Okay, so sister chromatid is the one that moves towards the opposite pole, okay, away from the metaphase plate. So what you're going to see here after anaphase 2, you're going to proceed to the telophase 2. Okay, so we have 2 cells from cleavage furrow. So nuclear envelopes start to reform. Nuclear envelopes start to reform. And you're going to get the blue one here for the first cells, and then green. Okay, this side you get the blue with a little bit of red, and then you have the green with a little bit of orange. Okay, so another cells. So I click furrow. Formations of the nuclear envelope take place. So here you have the red color, the blue, then the orange color with the green. Then you have the orange color and also the red color. Okay, so from the telophase 2, you enter into the cytokinesis. After cytokinesis, then what we're going to get here will be 4 cells. 1, 2, 3, 4. So this one will be the blue color, blue, a little red, red. Oops, sorry. Red, red. So a little blue. Can you see that? So green, green with a little bit of orange, orange with a little bit of green, and then it's orange. So you can see that we get all different kinds of the cells, different combination. We see that it's different combination. So now go back to this anaphase 2. You go back to anaphase 2. If I ask you number of chromosome, now this is a little bit question always asks the number of chromosome, but in anaphase 2 they actually refer to sister chromatid. Are you clear? This is the thing that students always confuse. When it comes to anaphase 2, question always asks you, okay, question asks, what is the number of chromosome per cell? But in this case, even though they use a chromosome, they refer to the sister chromatids. The answer is 1, 2, 3, 4. How about the number of DNA? Also equal to 4. Okay, for anaphase 2. How about telophase 2? Because we haven't started cytokinesis yet, so same thing, number of chromosome, which they actually refer to the sister chromatids. So in this case, also equal to 4. Number of DNA per cells also equal to 4. But after cytokinesis, can you see that number of chromatin? Can the step per cells now equal to 2? Number of DNA per cells also equal to 2. So that's what we have completed the whole meiosis process. Okay or not, guys? So just a quick revisions and the signature events of each phase. So I'll go through again with you guys now. So I'm going to start with this. You can take a photo if you need. This is what I see, interface. So what actually happens during the interface. So the most important thing that takes place in the interface basically is the DNA replication and replicates semi-conservatively during the S phase of the interface. Okay. So then prophase 1, what you need to know here is you can see that. Okay. So what you're going to see from a prophase 1 here, so condensation of chromatin as usual, homologous chromosome pair up, and then chiral crossing over, which we're going to lead to the genetic variations. And after crossing over, we can see that they join at the chiasmata, forming the bivalence or tetrad. Okay. Then when it comes to metaphase 1, the entire structure we call a bivalence of tetrad aligned at the metaphase plate. Okay. And independent assortment take place, which will give us the genetic variations later. During the anaphase 1, this is the key items that you guys need to understand. During the anaphase 1, you can see that the whole structure here we call as the chromosome, not sister chromatids. So the shortenings of the spindle microtubule actually break the chiasmata. Okay, not centromere yet at this moment, not centromere, but chiasmata. Okay, so therefore the chromosome now move to the opposite pole away from the metaphase plate. Okay, so telophase 1, nothing special until cytokinesis actually take place. Okay, so meiosis 1, the idea of meiosis 1 actually to segregate the homologous chromosome so that we can form haploid cells. But the question here is the DNA or sister chromatids still remain double up. Can I see that? Still remain double up. So therefore, we need to continue the meiosis 2, where we're going to segregate the sister chromatids so that form haploid cells, okay, with only one set of DNA. Okay, so that's why we continue with the prophase 2. So always remember, when it comes to the meiosis 2, is similar to the mitosis, So at the end of the process here, you can see that we're going to get 4 different types or different combinations of the cell which are genetically non-identical. Okay, so but one thing that I want to emphasize here during the anaphase, we always say that shortening of spindle microtubule, highlight here, divides the centromere, not break the centromere, not the splitting the centromere, but the correct word to be used here is divides. Okay, and then sister chromatids will move toward the opposite end, away from the metaphase plate. So when the telophase 2 and also the cytokinesis completes, we'll be able to get 4 different types of the cells, and each of them have different combinations of allele. Okay, so with this, I stop the recording.